Well, welcome everyone. I'm Diane Newman. I'm the proud mentor of the star of the show today. And I'm going to begin with a very brief introduction, well, maybe not so brief introduction, to Lev, which is the tradition we have in the lab. And it's a particular pleasure to do this, knowing that Lev's parents and extended family members, soon to be effectively already, are all present in the room. And so I want to welcome all of you. I know this is a really important day in all of your lives and also in mine and Lev's. And I think it's fantastic that you're here. So this story of this young man, of course, begins in Russia and was continued when he and his family moved to Colorado at the age of 8. But I will focus now on the academic journey thus far, which began after high school when Lev went to the University of Chicago. And he graduated with a BA in biology there. And perhaps the most important thing I think he got from this time was an incredible relationship with his mentor, Aaron Turkowitz, whom I believe is on the video watching today. And this relationship has been so rich and so giving in so many ways and extending even throughout his PhD experience here that I just wanted to honor it because it's been a very important part of his development as a scientist. Now, we were lucky to recruit Lev here in 2017 to start in the biology program at Caltech. And of course, today he's defending. But he will go on continuing in biology for a postdoc at Stanford in Eleniè's lab. And through this arc, beginning as an undergrad and going forward into his postdoc, Lev will have traversed a large part of the phylogenetic biological world using the tools of genomics and genetics. He's someone who has a real curiosity about biology writ large, and it's reflected in the diversity of the organisms that he's studied to date. Now, it's often said that you can learn a lot from a person by initial impressions. And I actually formed some of my first impressions of Lev when I got to watch him in action as a student in the microbial diversity course, which Jared Ledbetter and I co-taught, and Lev was a student in that course our last year. Whoops. And so what was immediately apparent was that Lev was the kind of guy who liked to shake things up, like literally and figuratively, as you'll learn in a moment. But what was also very clear from day one was just how bright he was, how inquisitive. The questions that you would ask in the lectures at this course were always just startling in their sort of unexpectedness and depth of insight. And I thought to myself, boy, I'd be really lucky if I could recruit him when he showed up at Caltech, but I wasn't sure yet how that would go. And I was even more motivated to recruit him when I observed that he was an incredible challah chef. And so here you see, after a triumphant challah party at my small apartment at Woods Hole, I think there are a total of 6 beautiful loaves. Lev, the czar of challah, being celebrated for his gifts in the kitchen. Yet another early sign of something that would come to characterize Lev as a student was his whimsy, which was evident early on, as you can see in his parody of Rene Magritte's ironic painting. And, you know, his lack of inhibition of sort of, you know, deciding, yeah, we'll stick it to the system and make this graffiti here on a famous bridge in Woods Hole, which I believe it still is up to this day. Now, this tendency to be able to make a very clever connection is something that at Caltech wound up, you know, manifesting over and over again. And one of my favorite examples is Lev's early renaming of our micro morning seminar series, the Seminar, which was like a very brilliant little twist that is just one of many examples of Lev's ability to make acronyms in a very creative way. Now, early on in Lev's first year, he told me that he was taking impromptu classes on the west side of LA. And I thought, that's interesting. You know, I'm sort of skeptical that's going to be sustainable, but it turned out, well, it wasn't sustainable that long in truth. But what it did was give him, or amplify perhaps is a better way of saying it, talents that were already innate. And this came to manifest in dramatic ways, such as in a group retreat we had prior to the pandemic, where I asked every participant to be able to share something about themselves that they thought was meaningful. And Lev decided he would share his love of doing impromptu speaking. And so he wound up giving this extraordinarily funny talk based on slides that he hadn't seen at all that my lab put together. And as you can see here, everyone around the table is greatly amused. But this ability to be quick-witted and think on your feet is something that can extend in ways that are more serious, of course, in academia. Most importantly, in the realm of teaching. And Lev is a really gifted teacher, as I know that Justin, who's on his committee, is nodding his head. Sarkis Mazmani and I, who had the pleasure of having Lev TA for us, microbial genetics, would acknowledge that Lev was effectively the third teacher of the class. His ability to explain things lucidly, and that coupled to something even more important, which is, I think, his really genuine caring for the students to understand the material and work with them, was something spectacular, and which is going to predict, I think, a really impactful career as an educator. And you'll have an example of his teaching in just a moment. But I can't, transition to the defense without acknowledging something a little bit more serious, because this was also part of Lev's time here at Caltech. And that is, not every moment is always lighthearted. And it's important at times to be able to speak up, even when it's challenging under difficult circumstances. And Lord knows the past few years have presented a variety of challenges to us all. But as hinted in this nice image, Lev doesn't shy away from a confrontation when it's for a good cause. And he can make even the most formidable opponents eventually bend to his will. And I think that we really can actually thank Lev for some very substantive and serious advocacy here within the Caltech community that led to some very meaningful changes for our graduate students, something of which to be very proud. And in wrapping up, what I also want to say is, I think you're going to be treated to Lev as a scholar, and I could talk about that for a long time. But as a mentor, you get to see many sides of a student, not just their academic talent, and you get to come to know them as a person. And these personal characteristics are the things that last longer in your memory than the results they may get in any given year in their PhD. And I think one of the things that I will always keep with me is a real appreciation and a respect and a love for Lev's part, because I think someone who is so sensitive as you can be challenged in walking through a world that is as imperfect as ours is. And yet I think what allows one to navigate that is the ability to remain hopeful and to be able to utilize one's resources and reach out to others for support and to give support in return, which you've done really magnificently. And so I see this as something optimistic and boding incredibly well for you in the long run. Yeah. And I could not end this, Jana, without acknowledging the fact that a critical achievement in the past 5 years has been the meeting and the wooing of Jana. And it's pretty wonderful. This is a photograph in June 2019. I think one of the first times I might have met you, but I love this because it just captures this great happiness and joy between the two of you, and the smile that somehow manages to exist, through really easy, fun times and also very challenging times. And to have seen you navigate that has been a beautiful thing. So with no further ado, I will now introduce Dr. Strangelove and let the defense begin.
Thank you so much, Dan. I couldn't have hoped for a kinder introduction. Hopefully, I'll be able to reciprocate it a little bit at the end of my defense. I promise you, I've practiced this talk. It's not just extemporaneous. We
switched on the slides anyway.
Oh, thank you. Okay. So I'll be talking to you today about my discovery and then exploration of the biological mechanisms of a widespread phenazine oxidation. And hopefully by the end of the talk, this title will make sense. So I'll start by really zooming out and talking about the big picture of life on Earth. And if we look at it just in terms of what life is there and how much it weighs, we see something like this, where just looking in gigatons of carbon mass, life on Earth is dominated by plants largely, and we can think of them as pretty much the most successful life form that we have. But then we have all of the microbes. We have the archaea, the viruses, the bacteria, protists, some fungi are also microbial. And of these, bacteria are dominant. And so this raises the immediate question of, well, how does something so tiny, compete, persist with something as successful as plants, for example? What does it mean to be so small? And so I'm going to focus on bacteria for the rest of the talk. So when I talk about bacteria being small, they're very small. So if we compare them to us, to a human being that weighs, say, an average of 100 kilograms when an adult, and compare that to the largest animal that's ever lived, the blue whale, the difference there is roughly 1,000-fold in weight. But if we compare this to bacteria, where here, for just a sense of scale, these little pores are 1/5 of a millionth of a meter big, we see that the difference in mass between us and them is 17 orders of magnitude, which is like an inconceivably large gap. And so when you're this small, you don't really know where you are. There's no way for you to really distinguish whether you're on land or at sea or whatever else. You just know your very local environment. And so the bacteria that somehow manage to colonize and grow and thrive on plants and in soil and animals, or just in the environment, do so by way of their diversity, in the sense of some bacteria are adapted to some environments, not others, or individual adaptability, where a given bacterium can modify the way that it behaves in order to survive no matter where it ends up. And so the natural question here for me is, how does a bacterium manage its natural environment or its environments? Now, to answer such questions, we have to think about how we study bacteria usually. And this here again, we kind of bump up against the issue of bacteria being very small. If something is, infinitesimal in mass, you need a lot of it in order to work with it. And so the general approach is to try to grow a lot of them quickly. And so if we take the kind of usual workhorse bacteria that we have in the lab, and we give them everything they need, they'll divide roughly every 30 minutes, and we can plot this. So here I'm going to have the mass of the bacterial culture, starting with a single bacterium over time and hours, and this y-axis is on the logarithmic scale, so the exponential growth will appear linear. So we've all become very familiar with exponential growth over the past couple of years. So we know how quickly things can change, but to just illustrate the significance of this, starting with a single bacterium, over the course of about a day, you're going to end up with a couple grams of biomass, which is equivalent to several sugar cubes. But then in just 3 or 4 days, that bacterial culture will have the mass of the whole Earth, and in less than a week of the entire observable universe. And that clearly doesn't happen. So this means that this way in which we normally work with bacteria in the lab isn't actually representative of what happens in nature, because the way that we try to treat them isn't sustainable in nature. So the relevant context for trying to understand how bacteria manage the environments is when they're growing slowly or not growing at all. And the question then for me becomes, how does a stressed bacterium manage its environment? So what I've come to learn as, I started in the microbial diversity course in 2017, and then in Diane's lab here, is that bacteria manage stress by building molecular tools. And there's a vast diversity of these tools. So one example are things like siderophores, which are molecules that allow bacteria to kind of steal away iron, whether it's in the context of an infection or in the context of being at sea and trying to grab water out of the iron out of the water and compete with other bacteria around it that way. They can also make antibiotics. So they can try to kill off their competitors by secreting toxic compounds and survive themselves and kind of win out over others. They can also rely on their numbers. So there's a phenomenon called acorn sensing, where bacteria make these signals that indicate to them when they're at a certain concentration that they have enough neighbors around them to do something together. And also, they can make something that's called electron shuttles, which is a way for bacteria to move electrons around in their environment and thus tune how they interact with it. And this was the thing that really caught my fancy, because just the idea that something like a bacterium can use electricity, which I thought was a purely human invention, was really fascinating to me. So let's think of life then as a kind of electron flow. And what I mean by this is if we take ourselves once again and think about what allows us to live and to metabolize, we can see it as, processing things like sugar into carbon dioxide and concomitantly breathing in oxygen and transforming it into water. And in doing so, we're actually doing kind of the equivalent of what would happen if we just lit sugar on fire, because it would burn, it would release energy in the form of heat. But what we accomplish as living organisms is to slow down that process and let it happen stepwise, so that at every interval we can conserve some of that energy rather than let it dissipate as heat. If this were the only thing that existed in the world, we'd run out of sugar and we'd run out of oxygen and life would cease to exist. And so we have our friends, for example, like plants, that can do the reverse. They can take carbon dioxide, make it back into sugar. They can take water, make it back into oxygen with the help of power from the sun. And then the cycle can continue with other organisms and life on Earth is sustained. So now if we abstract this away from the specific organisms that might be doing this, we can then trace what the electrons are doing in the process. So here, electrons are going from the sugar to the oxygen to form water, then from the water to the carbon dioxide to form sugar again, and then the process repeats. And we can then continue abstracting this away by forgetting about specifics of sugar and oxygen and just call it an electron donor and acceptor. And then we see that the donors and acceptors flip according to the metabolism at hand of what the organism is adapted to do. And so then if we look at a given organism doing its one thing, really what's happening is it's taking a terminal electron donor and a terminal electron acceptor, and then reducing the acceptor, oxidizing the donor, taking the electrons away or putting them on, and producing waste. And that waste can be food for other organisms, but as far as this one's concerned, that's all it cares about. And so then all of what life's mysteries basically happen in this context. This is reminiscent to me of a quote that I really like from Albert von Jahn-Georgi, that life is nothing but an electron looking for a place to rest, which when I first heard it, I found overly fanciful, but now I'm very much on board. So now I'm going to focus in back on the electron shuttles, these molecules that help break up the flow of electrons in a stepwise way. And I'm going to focus specifically on phenazines, because these are the electron shuttles that the whole lab is largely focused on. And so these can be thought of as life and death electron shuttles. And I'll illustrate to you what I mean. So if you imagine bacteria growing as a colony on the surface of an agar plate, which is something that we commonly do, but we could also imagine this on another surface, like on a pipe or in a wound. These bacteria, if they're respiring oxygen, will consume it very quickly. In fact, the bacteria that are on the surface of the colony are going to consume it faster than the oxygen can diffuse inside. And at a depth of even so much as 50 microns, the internal environment here becomes anoxic. And so if you're a bacterium that wants oxygen and you find yourself in this environment, you're very displeased because you have nowhere to put the electrons that you have in your metabolic waste. But as I mentioned, the bacteria can make their own molecular tools. And in this case, they can make phenazines and dump their electrons onto the phenazines themselves. One moment, I'm just going to make sure my cursor is always visible. Okay. So this bacterium made its phenazine and it put electrons onto it instead of oxygen because oxygen is not available. And then the phenazine can diffuse away to an environment that is more oxygen rich, react with that oxygen abiotically. Oxygen is a very reactive compound, and then kind of diffuse back, and the cycle can continue and the bacteria are supported. But in this process, you create superoxide, which is a deadly compound. It disrupts all forms of life if it's there in excess. And so while the bacterium here is benefiting, those neighbors of it that created the situation in the first place might actually be hurt. And if there's a problem of kind of collective action here where you're trying to support the whole population, this is a bit of an issue. And so with this in mind, we can postulate biological phenazine oxidation rather than abiotic with oxygen. So this would be a shift from a schematic like this to one like this, where instead of relying on diffusion and the reaction with oxygen directly, you can have a different bacterial partner that oxidizes the phenazine, resolving the issue of toxic superoxide production. And then that bacterium can respire oxygen or something else, whatever it needs, but you don't necessarily get the toxic intermediate. So I'm going to focus in on a specific phenazine out of all of the myriad phenazines that are out there, because it's the most basic one. So phenazine 1-carboxylic acid, which I'll refer to as PCA from now on, but that's not to be confused with principal component analysis, is produced by many, many bacteria. So this is a technical phylogeny that shows the relationships of all the different bacteria that make it. But the conclusion to draw from this, if you're familiar with any of these clades, is that PCA is going to be produced wherever there are root-associated bacteria, and then in other environments too, but pretty much where there's plant life, there will be phenazines, and as I showed you at the very beginning, there's a lot of plant life out there. So I'm schematizing this phenazine as these 3 hexagons, and that's because its real structure is very similar to that. It's tricyclic with these aromatic rings with 2 nitrogens in the middle ring, and then this carboxylic acid group decorating it. And what it can do is it can accept, oh, hold on. My computer is lagging a little bit. Hello. Okay. So it can accept 2 protons and 2 electrons to be reduced. And those protons and electrons land on those middle nitrogens. And what's really cool about this is that when it's reduced, it becomes brightly green. And this is very useful, both because you can just visually see whether it's reduced or oxidized, but it also has a corollary effect that is highly fluorescent. So you can measure it in a specialized equipment that way too. Then the 2 electrons, 2 protons can be lost and the thing can be reoxidized. And this cycle can happen ad infinitum without any wear or tear on the molecule. So the question for me becomes, is biological PCA oxidation possible? We kind of postulated that it'd be nice if it existed, but that doesn't mean that it should. And then following this, I'd want to know where and how we could actually find it, what the consequences of it are, and how it works. So to address the question of whether biological PCA oxidation is possible, I'll give kind of a brief primer on thermodynamic thinking and metabolism, and I'll try to make it as accessible as possible. So when I talk about PCA reduction, I'm talking about a chemical half reaction. And the reason why it's a half reaction is because it's not balanced. There's no conservation of mass here. Out of nowhere, we get 2 electrons and 2 protons that the phenazine grabs, and then it's reduced. And we can actually measure or calculate the potential energy of this half reaction. So this is measured in volts. And the circle and apostrophe indicate that it's under standard conditions, meaning room temperature, 1 atmosphere of pressure, neutral pH, and everything, all the reactants, all products at 1 molar concentration. In order to get a full reaction that's actually balanced, we need to partner this with a different half reaction. So in this case, I'm showing a reduction of nitrate. And here I'm using nitrate as an example because after oxygen, it's one of the most favorable electron acceptors out there, but it's much less reactive than oxygen is on its own. And so if we're looking for PC oxidation, this is a good electron acceptor to use. And so we see kind of a similar thing, that this is a 2 electron, 2 proton process, and that the potential energy of this reaction is now in positive millivolts. And the way to interpret this is that, as written, the second half reaction is favorable, the first one is not. But we can put them together in the favorable conformation, where the reduced PCA reacts with nitrate and becomes oxidized, and the nitrate is reduced to nitrite. And then by subtracting away this potential energy from the first half reaction from the second, we get that this has 549 millivolts of potential energy under standard conditions. But the thing that we care about when it comes to reactions, in terms of determining whether they're possible, is what's called the Gibbs free energy. And if the Gibbs free energy is negative, then the reaction is favorable. It can actually happen. It's not against the laws of thermodynamics. And so if we, I jumped ahead a little bit. I'm going to denote in color the reactants and products because they're going to come up a little bit later. So we can relate the potential energy of the full reaction to the Gibbs free energy with this equation, where these 2 are just coefficients. We get a negative number. And so this tells us that under standard conditions, this reaction is favorable. But biology doesn't happen under standard conditions. And while in the lab, I can control for the pH, and I can control for the temperature, I can control for the pressure, I can't really control for the concentrations of the products and reactants, because they're going to be used by the cells. They're going to change dynamically over time. So the best that I can really do is ask, what happens when the ratio of the products and reactants changes? And we can relate this standard Gibbs free energy to the true Gibbs free energy of the reaction with this relationship with the reactants and the products. And so basically I can ask the question, with what ratio of products to reactants will this inequality be less than 0? And what I find if I rearrange this and do a little bit of arithmetic is that the reaction is favorable if the ratio of products to reactants is less than 2.1 times 10 to the 20th. And this is a massive difference. This is greater than the difference in mass between us and bacteria. Effectively, it means that this reaction will proceed up until we can't even imagine it proceeding further. So it's very favorable, thermodynamically speaking. So now that we know that it's possible, the question is how do we find it? And specifically in the context of microbial chemical ecology. And a way of finding life that you don't know whether it exists or not is to do what's called an enrichment. Basically, create conditions that will be favorable for it to grow according to first principles, and then see what happens. And so this was started by Yanon Baron, who was a student of the microbial diversity course, and Scott Saunders, a former graduate student of our lab and a TA at the course at the time, who went out and at 3 different locations in Cape Cod took topsoil and asked whether there would be phenazine oxidizers there. And so the way to do this is to set up conditions that will give you the best likelihood of success. So here we have a stoppered vial with an anoxic atmosphere. So there's no oxygen there to abiotically oxidize the PCA. We have a reduced PCA minimal medium. It's green because the PCA is reduced, and the minimal medium just means that we know everything that's in there. It's completely defined. And then it includes nitrate. As I showed you with nitrate, this thing should be possible. And then there's the soil inoculum on the bottom. And the thing to do is set it up and then wait and see if you lose the green color. And if you do, that's evidence that the PCA was being oxidized. And then you can take an inoculum from this first enrichment, put it into a fresh tube and wait and see if it oxidizes the PCA. And then you do it again, and you do it again, and you do it until you're convinced that you're passaging something that's alive and not something weird that you got out of soil. And just to illustrate to you what this looked like when I had it in my hands already here at Caltech, here are 5 tubes. The leftmost one is a negative control, so there's no inoculum there. And the other 4 are enrichment cultures that we got from Woods Hole that I was passaging. So when I first started, everything is green. In 3 days, a couple of them are starting to turn clear. In another 3 days, they've all turned clear. And the loss of volume here is because I sampled from them because they're going the fastest. And I just want to make a quick aside to say that letting biology lead the way in this way, when you're doing an unbiased enrichment, can lead to some unexpected discoveries. One of the things that I came across during my PhD, and this John and I were looking at this together, is I got isolate out of the enrichment culture that under certain conditions made these dynamic waves in very thin colonies on agar plates. And so this is a video of a time lapse over 10 minutes, watching how some of these curls in this colony change over time. And I have no idea what's going on here. The strain is in our freezer. And if you're starting out here, or if you want to bring this into your lab, please feel welcome to hit us up, because we'd be happy for someone to work on it. But despite the allure of these wavy colonies, I had to refocus to get my PhD done. And so for the rest of the talk, I'll be talking about the PCA oxidation. So as I mentioned, I can use PCA fluorescence as a proxy for whether it's reduced or oxidized. And I can do this in high throughput in a plate reader. So this is a device that lets you put in plates with 96 wells. And so each little well can be an experiment, and you measure them all at once. And here I can measure fluorescence over time. Here it's just the fluorescence in arbitrary units. And I'll be showing you how one of the enrichment cultures behaved over time. This is one of the first experiments that I did with it. So I'm going to compare the enrichment culture given only reduced PCA, given fumarate, nitrite, and nitrate. The reason why these 3 things are here is that the nitrate is what we provided in the first place in the enrichment. The nitrite is something that appears when you reduce nitrate, and fumarate was basically a control for a different thing that organisms can respire. And so what this enrichment culture does is, if you only give it reduced PCA, the fluorescence doesn't change, which is indicative of no oxidation. And if you give it fumarate, the fluorescence doesn't change. If you give it nitrite, it oxidizes a bit, but with nitrate, you get a very rapid loss of fluorescence down to the kind of floor of detection, meaning that all of the PCA gets oxidized. And after a certain amount of time, I found that they actually re-reduced the phenazine, which I won't talk about today, but it's basically a consequence of running out of the terminal electron acceptor. So then I could compare this to another enrichment culture that we got in parallel that behaved very similarly, and to one of the isolates that I got out of it. So this is pure isolate, completely isogenic bacterium that pretty much recapitulates the dynamics of the original enrichment cultures. And so I focused on it, And together with Ynon and Scott and Diane and Jared, we published a genome sequence announcement of it. And it required a whole genome sequence analysis because, as it turned out, it was very highly similar to other species. I identified it as a strain of something called Citrobacter porticolensis, which was first identified in 2017, actually, in the year, or at least it was first published in 2017, the year of the microbial diversity course when we were there. And an interesting aside to the kind of microbial ecologists out there, all of these organisms have basically 100% identical 16S RNA. And so it wasn't sufficient to actually identify what species it was without a whole genome comparison. So having this organism in hand, I needed to kind of systematically go through the potential mechanisms of how PCA oxidation might work. So one formal possibility that's pretty much ruled out, but we still need to consider it, is that there is an abiotic reaction with nitrate. The reason why I say it's pretty much ruled out is because I showed you the negative control of the tube that stayed green while the others oxidized. But nonetheless, a formal possibility is that somehow, some way, the PCA is being oxidized by nitrate directly. A slightly elaborated model is that the PCA is still being oxidized abiotically, but by nitrite, which is produced by bacteria as they respire nitrate. So this requires microbial activity, but it wouldn't have anything actually to do with the bacteria themselves. The third option would be that the bacteria are directly coupling the respiration of nitrate with the oxidation of the PCA. And the fourth possibility, which is a slight elaboration, is that the nitrate works, but it's not necessary. Maybe it's any form of anaerobic respiration that works for this, with any terminal electron acceptor. That's what TEA stands for. Or maybe specific ones, but maybe it's just more general than nitrate itself. And so to pinpoint this, I'm now going to show you more summary statistics rather than myriad curves. And what I'll be plotting for you are the maximum PC oxidation rate in micromolar per hour for the different cultures incubated under different conditions. And so I'll be comparing my strain of the Citrobacter to an abiotic control, as well as to a couple other bacteria I had in the lab. E. coli is just a standard point of comparison for a commonly used bacterium. And then we use Pseudomonads as bacteria that actually make phenazines, but in this case, they were mutated not to make them anymore so that there weren't any spurious phenazine effects. And so here I'm showing you the situation with just reduced PCA, and with just reduced PCA, none of them oxidize it. The basal oxidation rate is near zero. You know, some days there was more oxygen in their aerobic chamber than others, so there could be a little bit going on. But effectively, nothing happens if you don't have a terminal electron acceptor. Then if I compare this to the situation when I provide the reduced PCA and nitrate, there's a stark contrast where the abiotic control remains near zero. My isolate oxidizes PCA very quickly. And then a couple of the other organisms oxidize it too, but not all of them. So this means we can rule out this thing that was unlikely anyway. There's no abiotic reaction with nitrate that contributes to this appreciably. So now we have this elaborated model where it's not the nitrate, it's the nitrite, but the nitrite comes from the bacteria. So here we can compare the isolate that I have to the abiotic control. And if you squint, you can see that the abiotic control is oxidizing. It's a low rate, but it's above zero. However, if you compare that to this rate of oxidation given nitrate, there's no way that this low oxidation rate could explain that activity. And the reason why I say this is because the concentration of nitrite here is equivalent to the total concentration of nitrate there. Comparing this to the other strains, it looks like two of the Pseudomonads actually seem to like to oxidize PCA with nitrate. But overall, we can say that, at least with my isolate, this model isn't the dominant contributor to what's going on. So then we're left with this idea that the isolate is actually coupling PC oxidation to nitrate respiration. And to compare it just to the elaborated model with fumarate, here my isolate is the only one that oxidizes PCA. And so at least for it, we can say that this expanded model where anaerobic respiration that's not strictly nitrate can drive PCA oxidation. So I see that nitrate stimulates PCA oxidation, and I wanted to show that a bit more dynamically. And I did this in this bioelectrochemical setup where I had my bacteria oxidizing PCA with nitrate, but also an electrode that continually reduced the PCA. And so long as the bacteria are oxidizing it, there will be a current that I can measure. And the way that this looks in real life is like this. Inside one of our anaerobic chambers, there are wires everywhere and make me feel like a mad scientist. And the result is if I incubate the cells, don't give them any terminal electron acceptor, no nitrate, the current is basically within the noise. This is on a logarithmic scale again. And so these big differences are actually kind of artifacts of the scaling. If I provide them nitrate to start with, they can hold a current of roughly 120 microamperes for around half a day. And then the question is, well, does the current drop because the cells die or because they run out of nitrate or what? And so I can check this by spiking nitrate back in. And so here I spiked nitrate every 24 hours. And as soon as I did, the current rose again, which means that they ran out of nitrate to use. And eventually their metabolism seems to slow down enough that they stop running out of nitrate. And so the current just persists. And if I spike into the nitrate every 12 hours, the current is never lost. So it really seems like nitrate is the thing that's allowing them to do this. But I also saw that PCA oxidation stimulates nitrate reduction. And I saw this by tracking the concentration of nitrate and nitrite over time. This is using a technique called ion chromatography. And basically, if I compare a situation where I provide no phenazine, when I provide only oxidized phenazine, or if I provide the reduced phenazine at the start, I see a real difference in how much nitrate the cells can consume, where the cells that were given reduced phenazines consume the nitrate faster. And this is reflected by the production of nitrite. So all of the nitrate that is lost here appears as nitrite over time, and that conversion is stoichiometric. So the nitrite just stays there and doesn't really do anything. I also found that nitrate-driven oxidation appears to confer a survival benefit. And this is again in this bioelectrochemical setup, where if I compare the colony forming units, basically how many viable cells there are in the culture over time, while the cells have access to the PCA and the electrode, they don't die over the course of 4 days. But if I remove the PCA and give them only an electrode, this is a mistake, I should have put in the X over here, then after roughly 3 days, the cultures start to die. So there's something here where this process is actually doing something for the cells. And so at this point, we arrive at this kind of self-contained PCA redox cycle that the cells can do. The conventional logic prior to this work was basically that the cells can do what they do during respiration, meaning take internal electron stores, reduce the terminal electron acceptor, and this way reduce the phenazine too. And then, as I showed you, there is an abiotic oxidation that can happen with nitrite, though it's not huge. But there is the reverse reaction happening at the same time, where the cells, while reducing the nitrate, are oxidizing the PCA. And in this way, the cells are catalyzing a complete redox cycle here. So for the rest of the talk, I'm going to try to give you some insight into how this actually works. And so again, we need to talk about potential mechanisms. The bacteria that I'm working with are kind of analogous to mitochondria in that they have an inner membrane, a periplasm, which is called something else in mitochondria, and an outer membrane. But this anaerobic respiration is happening basically at an enzyme complex at the inner membrane, where it's taking the terminal electron acceptor and reducing it by taking electrons from endogenous electron shuttles called the quinols. So my organism has ubiquinol, dimethylmenaquinol, and menaquinol that when oxidized are called ubiquinone, dimethylmenaquinone, and menaquinone. And so when thinking about where the PCA can be contributing electrons, it can be either here, kind of more directly giving electrons to the enzyme complex, or by regenerating this quinol pool. Either way, the electrons get delivered to the terminal electron acceptor. So we have these two categories, this terminal reductase interaction and quinol regeneration. And the way that I can start to distinguish them is by getting rid of the terminal reductases, or getting rid of the quinols, or getting rid of both, and kind of asking, how does this break the process? So here I got very lucky because I needed to genetically modify this organism that we got out of soil, which is not at all guaranteed to be easy. But I stumbled on something reminiscent of a quote from a different Albert, which is that from the elephant to the butyric acid bacterium, it is all the same. And in my case, what this manifests as from ecology to Citrobacter portuensis, the tools all work. And by tools here, I mean homologous recombination and oligo-mediated recombinering. It all kind of came together very nicely. And so what I was able to do is, again, with the scheme of knocking out the terminal reductases and looking at the maximum PCA oxidation rate, comparing the wild type to the abiotic condition, again, I see that there's variance, but the rate of PCA oxidation is quite fast. I can compare it to the single knockouts of the terminal reductases. This organism has 3 different ones. And here, some of them have an effect, some don't. Then I can look at all the double knockouts of all the pairs of combinations. And now I'm seeing that I'm really starting to reduce the PC oxidation rate. And finally, I can knock all of them out and pretty much abolish it. And so what this means is that any one of the terminal reductases can drive this process. But the process does require the terminal reductase activity. So then I can knock out the quinones. And here it's a similar story. I can get rid of the menaquinones, dimethylmenaquinones. I lose some of the activity. I can get rid of the ubiquinones. The activity isn't very much lost. And if I get rid of all of them, most of the activity is lost, but not all of it. So the oxidation involves the quinol regeneration process, but does not actually require it. And then if I then start getting rid of, on top of this, the pairs of the different terminal reductases, I can see that the PCA oxidation still persists. And so any one of the terminal reductases, even when there are no quinols around, can actually drive this process. So the implication here is that there's a very general mechanism for biological PCA oxidation. What I drew here in the beginning of this section wasn't specific to nitrate. It's just about a terminal electron acceptor. And so I've done this with more molecules, but just to give you an example with fumarate, one of these alternative terminal electron acceptors, I can again compare PCA oxidation with fumarate, with my wild type, or with the abiotic control. I can compare it to a fumarate reductase knockout, which I got with thanks to my rotation student, Alan. And then I can compare it to the quinol knockout. And it's a similar case as with the nitrate reductase, where if I get rid of all the quinones, some of the oxidation rate is gone, but not all of it. And then I can complement this back. So I can reintroduce the wild type fumarate reductase and kind of rescue it from this low point to here. I can reintroduce dimethylmenaquinones, menaquinones, and get back the activity from here. And the ubiquinone complement doesn't really seem to help. And so basically, the picture here is that we're gaining an understanding of how this works, and we can apply it in a general framework. I'm going to skip ahead a little bit because I'm running low on time. No, I don't want to. Okay, so to give you a narrative recap, what I hope I've shown you today in a compelling way is that we were able to kind of postulate that a metabolism is possible when that hadn't been observed or appreciated before, and then go out into nature and actually find it, which in itself is a very cool thing. And then systematically, just kind of thinking through the different possibilities of what might be going on, I was able to rule out the abiotic models and focus in on the ones where PC oxidation is actually coupled to anaerobic respiration. And in the course of this, found that a single bug, a single bacterium can at times oxidize or reduce PCA depending on the exact conditions that it finds itself in, and that under certain circumstances, it actually appears to be helpful for it to do so. Then I zoomed in further to try to get at the mechanism of how this works within the cell and proposed two different models how it might work, and both of them seem to contribute. And so the implications of all of this are that PCA oxidation occurs and should occur wherever there's PCA and anaerobic respiration happening. And I kind of alluded this in some of my charts, but basically any bacterium that I've tried in the lab to get it to oxidize PCA did oxidize PCA. And so, so long as it had what it needed to anaerobically respire. Another consequence of this is that PCA oxidation changes its surrounding environment. I saw this in the sense of nitrate being depleted faster and being converted to nitrite. And this is something that can be a determinant for soil fertility, so that is important. But also that, just kind of put myself out there, it's possible, given what we know now, to go out and postulate a metabolism and within 5 years, find it and actually figure out the broad strokes of how it works. And I think that there's a whole world out there that people can explore. Okay, so with that, I'm going to move on to the most important part of the talk. And I can't acknowledge everyone here. I just don't have the time. I've written out 4 pages in my dissertation that I welcome all of you to read when it's available. But I wanted to start, of course, by thanking Diane. You're a very important person in my life and have been for a long time. And I know that you'll continue to be. And really, so much of my life and how I've developed and how I've grown over the past the past 5 years are thanks to you. So thank you. And that goes to all the iterations of the lab over the years. It's very strange to have watched a whole turnover of the lab as I've been here, but it's very much true that everyone who has been in the lab, whether it was for a long time or a short while, have really helped me and contributed to my well-being and happiness here. And it's also very much true that the friendships that I formed while here are ones that I hope will last for the rest of my life. And it makes me very happy that a lot of the photos that I have are of us in nature, because I feel like that has been a lot of when we've connected the most. Some of the friends that I've made too aren't from Caltech. So here are 2 from the microbial diversity course. I'm also incredibly grateful for friends I've met through Yana, because I feel very close to them, and it's a real joy to be able to move soon to the Bay Area and to actually have a community there to move to. I also really deeply want to thank my parents for a lot of things. My life is as it is because of them and because of their choices. And a lot of the things that I love and appreciate most in the world, like foraging for mushrooms, definitely comes from them. And so I'm endlessly grateful to the life that I've had thanks to them. And I'm also very grateful to grow and join a family too here that have welcomed me so warmly into their lives and into their home. And of course, I have to acknowledge Yana, Yana herself. Yana, I love you. You've helped me through both vague and extremely detailed unrest. And I'm very excited to continue developing our lives together with our kitties and whatever else life brings. So thank you, everyone. Right. I'm happy to take questions here or on the Zoom chat. I'm going to try to repeat the questions as best as I can so that people on Zoom can hear them. But if I forget to, please remind me and I'll repeat them. Yes, Georgia.
I'll start. I've always wondered what your vision is for the ecology of this process. As you point out, a lot of bacteria generally can do this oxidation, but clearly your isolate doesn't make phenazine, as I understand. It's one of the most efficient. So do you anticipate finding these efficient oxidizers near reducers, or how do you think this will work?
That's a very good question. So the question is about my intuition about the ecology of PCA oxidation in nature and whether really fast oxidizers will be partnered with phenazine producers. I guess I don't have a very good sense because I feel like what you'll see is the kind of compounding and the addition of all of the bacteria that are there that are anaerobically respiring. So if you have a phenazine producer or phenazine reducer, like Pseudomonas aeruginosa or something there, and then next to it are things that respire nitrate and things that respire fumarate and things that respire DMSO. They'll all be contributing to this. And so if it is the case that there is a real advantage to oxidizing the phenazine, then yes, I could imagine that there will be organisms that kind of evolve over time to do it faster to outcompete the others. But if it's something that's largely just a side effect of the fact that electrons are flowing through the electron transport chain and through the quinols, then I don't think there will be much of a selection there. So I think it really depends on whether we can pin down where there might be a fitness effect. Yeah. Yeah.
This is somewhat related, actually. Right? I was wondering if you had a sense of the kinetics and maybe the oxygen concentration at which the biological activity would outpace antibiotics.
Yeah. So the question is whether I have a sense for the kinetics or whether at what concentration of oxygen these biological processes might win out over oxygen. It's hard for me to say. So in terms of the kinetics of PCA oxidation, I can look at, for example, the current generated over time, and I can't do it in my head right now, but saying that, okay, they're oxidizing PCA and generating 120 microamperes of current, gives you how much charge is going, and that tells you how many electrons, and you can convert that to the amount of PCA per unit time that's being oxidized. And I've done that calculation, I just don't remember that right now. But in terms of competing with oxygen, I really think that the oxygen needs to be depleted because different phenazines react with it at different rates. So PCA is slower than piocyanin, for example. But still, I once tried a quick experiment where I didn't let all of the oxygen get purged from my solution to see what if there's just a teeny bit of oxygen, I can watch cells use it to oxidize PCA rather than abiotically, but the abiotic control is still much too fast. So in that, I'm glad that was closed. So in that sense, I really think it needs to be anoxic. Renee.
Just like out of pure curiosity, but I didn't know about the waves thing.
Yeah.
Do you see that at all stages or only at the end when it's like?
No, so they would do this only after I had so the question is about the waves propagating from the colonies. And when I see this, I would see it only after I subjected them to the bioelectrochemical reactor for like a week and then plated them on minimal medium. And then they're like, and I have no idea. I have basically no idea. Like there's a possibility that what's happening here is that the colony is so thin that some kind of water evaporation is driving some weird materials dynamics. It might be that the bacteria are actually changing orientation in some pattern. I really have no idea. Okay, I think there's a question on the chat. I can't tell if it's a question. It's a comment. Okay, I'll talk to James later about the comment because I can't read right now. Yeah.
Is this happening in our bodies in an important way?
So it might be, but mostly in the context of chronic infections, if it is. So the question is this happening in our bodies? One of the contexts in which we study phenazines in the lab is in the case of cystic fibrosis, where one of the dominant pathogens that tends to arise is Pseudomonas aeruginosa, which creates phenazines, and that helps its pathogenicity, helps its survival. And what's very interesting about that environment is that kind of paradoxically, even though it's in the lungs, there's actually very little oxygen in the sputum. What you have, though, is you have various nitrogen oxide species. You have nitrate, you have nitric oxide that comes from macrophages in part, and conceivably the bacteria in that infection could be using those external electron acceptors to oxidize the phenazines that the Pseudomonas makes. But as far as I know, for example, just in our gut microbiome, there aren't really phenazine producers that are involved. There's a question in the chat from Scott. What about other phenazines? If this oxidation process is widespread, does it also work with molecules besides PCA? The answer is yes, but I haven't really looked into it. The very first indication for this process of phenazines actually came from a different graduate student who defended in 2009, I think. This is Alexa Price-Whelan, who was working on phenazine reduction. This is a different phenazine, and kind of noticed that in certain mutants, the phenazine remained oxidized it normally was oxidized, but stopped being oxidized in certain mutants. And you can tell this because phenazine is blue when oxidized and colorless when reduced. So it's kind of the reciprocal of my situation with PCA. So I was able to do just a brief pilot experiment with some reduced phenazine and reduced phenazine 1-carboxamide. And I saw that the cells seemed to oxidize it, but I never really followed it up because of solubility issues and being pressed for time. But I think that it does work for other phenazines. John.
So the mechanism that you present at the end seems like it's entirely encapsulated within the membranous components of the cell's electron transport chains. Do you think that the rest of the metabolism of the cell is in any way affecting this process? Because your electrochemical data showed that after day 3, I think, that stimulating with more nitrate does not produce current anymore. Would you care to speculate a bit about what you think that is?
Yeah, let me just pull those data up so that we're talking about the same thing. Beep, 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 beep. All right, here we go. You're talking about this top right plot, right?
Or no, the bottom left. I think I did this to memory. So
Yeah.
The result is that after the second spike, they continuously generate current.
Yeah, they do, because I think they slow down enough that they don't run out of nitrate.
Yeah, I see.
Yeah.
So do you think this process, I guess going back to the original point, do you think this process is completely independent of the cell's metabolism? Do you just need the enzyme around?
Yeah, well, so the question is, given how general the process is, and that it seems, at least the way that I presented it, constrained to the membrane, does it depend at all on what's going on in the cytosol and the metabolism, right? I think it does, but I can't really say how, in the sense that if one of the dominant factors is flow of electrons through the quinol pool, if the metabolism in the cell is such that it's just keeping all of those quinols reduced all the time, and there's no room for the PCA to put electrons onto there, that should affect it. But I really can't speak to, what might be happening, for example, for an organism that makes the phenazine, while that phenazine still is in a cytosol, how that might interact with other components of the metabolism too. I think there's just a lot out there that I'm not equipped to speak to. There's another question from the chat. Okay, can your Citrobacter isolate respire other anaerobic TAs aside from fumarate, nitrate, nitrite? And if so, do these TAs yield PCA oxidation? Yes, thank you for asking. My bug can anaerobically respire nitrate, fumarate, DMSO, and TMAO. DMSO is a common lab solvent. TMAO is an important marine metabolite, which is responsible for stinky fish smell. And with any of those, it oxidizes PCA. I just didn't have time to get into it, but I knocked out those terminal reductases. I knocked out I did all the same things that I did with nitrate and fumarate with those two. Yeah.
Yeah, to follow up on the question that you got there in the lower left. And that current is a time-based thing, right?
Yeah.
And so I don't quite understand how slowing it down, you're still getting the same level of current.
Yes.
So, yeah.
Yeah.
How are you able to-
So one of the things that I found is these bacteria seem to be very greedy in a sense. So if I give them nitrate and acetate, so like in all of these experiments, there's no other carbon source. They're just kind of persisting and not growing. If I give them acetate, they don't grow, but they consume all the nitrate super fast and then die. And in that process, the current of PCA doesn't change either. And so my sense is that there is some maybe maximum rate at which PCA can be coupled to nitrate reduction by these cells. And then if they're doing something else, like reducing the nitrate with the electrons from acetate, they run out of the nitrate and the PCA oxidation stops. And so here, I think what happens is once we reach this point of, I lost my mouse. Okay, here it is. Once we reach this point, the nitrate is being consumed by the cells slowly enough that they just don't run out of it, but also quickly enough that the PCA oxidation rate is still maximally coupled to it. That's kind of the hand-wavy explanation that I can give, but I think it would take substantially more experiments to actually nail it down.
Can you test that and then test it with explore that by asking how many electrons are actually
Yes.
used in that?
Yeah.
How
So,
much nitrate reduction?
yeah. So one way that I actually looked at this, which is still indirect, but quite interesting, is if we look at the difference of the nitrate reduction over these first 8 hours between the different conditions. The difference here is roughly 1 millimolar, but I provided only 200 micromolar reduced PCA. So if all of that PCA is oxidized only once, the most it could possibly contribute is 200 micromolar, but it does more. And so what seems is happening is the PCA is fully oxidized, contributes those electrons, then it gets reduced by the cell again, and then delivers those electrons back to nitrate. And that speaks to the model that I drew out here, where there's this concomitant process of the cells reducing the PCA and oxidizing it at the same time, so long as they have these internal electron stores. So something that I haven't done, which I would like to do, or would have liked to do, but well, somebody else will do it, is completely starve the cells first so that they don't have any internal electron stores, and then see how those electrons are delivered to nitrate and what the stoichiometry is and how that changes depending on changing concentrations. Okay. Okay, one more question from the chat. This is from Scott again. Maybe I missed this, but do you think PCA red can donate, this reduced PCA, can donate electrons to metabolism way upstream of the respiratory chain? Could you assess this by knocking out NADH dehydrogenase? I think it could. So I drew this in my model as just kind of directly regenerating the quinol pool, but there's no particular reason not to say that there might be intermediaries here. However, these are experiments that John has done. You can just mix together reduced phenazine and say ubiquinone, and in solution they react like that, and it's oxidized. So while it's formally possible that there are other steps here, it's not necessary that it is the case. But knocking out an NADH dehydrogenase would be a good way to check something like this. All right, shall we proceed? Any more questions?
Okay, then I think it's a good time.